Okay. So I was going over everything that I needed to get through for the exam next week. And it seems that there's no way we're going to get to them all, but whatever, that's fine. So just whatever we get through up until the end of Thursday is what will be on the exam. Which means that we're going to do more cytoskeleton next week, and that's just not going to be on the exam for the next exam. So that's how it goes. Okay. Exam 3 next week. I know it feels like we just had exam 2. It's that time of the semester. Everything seems to be going faster, and everything seems to be busier. Oral exam. Some of you guys have talked to me about it. Some of you guys have taken it. Some of you guys have found it does work for you. Some of you guys have found it doesn't work for you. If you'd like to do it next week, Wednesday's the day. So please let me know so I schedule you. Extra credits by Friday, 5 p.m. I keep getting emails saying, what about extra credit from last exam? I haven't put it on yet. I'm putting it on yet. I think I used 1 or 2 questions on the last exam, so you'll notice your grade go up as soon as I get a chance to put it down. If at the end of the semester you didn't see a change in your grade and you're sure I used yours, please email me because maybe I forgot. This week is open recitation, and we're going to do a drawing exercise. I just want to take a quick poll. Who likes the idea of open recitation and thinks we should keep things how they are? Who doesn't like it and wishes that we had some kind of more formal questions that you guys can go over where you all sit in class? Like, so take away the optional part of it, make it more of a normal recitation. Still, we could do practice problems, so who would rather do that? Okay, a few people. And then who thinks that we're not getting enough time to do papers and would prefer doing more primary research papers over open recitation? Okay, I got 1. Honesty is important. Okay, 2. No.
No.
3.
Since we missed all those recitations before, are we still gonna get in 8?
You're not, but we never do. I always have a goal of 8, but I always know that might not happen. So you always get to drop 1. Always get to drop 1. Yes. Sometimes I do formal reviews in lecture if we finish the material and I have an empty day. Yeah, and that's always really fun. But think of the open recitations as a time to ask me for those things. So like for the last exam, I got up there and I was like, okay, think of it this way, and we organized that. So if you come to the open recitation, you will get that. If you don't come, then. Okay, so it sounds like we'll keep things going as planned, and I'm going to figure out what to do next semester. Okay, take 5 minutes to write. So the topic of the day is nucleating proteins. What are nucleating proteins? How do they affect cytoskeletal dynamics? Discuss the 3 nucleating proteins in class, we covered in class, in terms of their substrates. I wasn't sure if we use the substrate in this context. That's why there's a question mark, but really their target protein, right? How they work, and the kinds of structures and cellular functions they are associated with. Certainly, their nucleating proteins make certain structures which are associated with certain cellular functions. So take your 5 minutes. Okay. Charles. So tell me, how did you approach this?
This whatever like Nano Chamberlain and ARP, they store the center for the elements to grow from. And Nano Chamberlain has microtubule, and the ARP is active. ARP goes to the cell membrane and adds to the negative end, I think.
Okay, so I heard gamma tubulin for microtubules, ARP for actin. They they create a nucleus so that subunits can quickly become polymers without that lag phase. What other nucleating protein? Yep. Formins. So that's one more. And so, what's the substrate? Substrate is, I'm not sure. What's the target of formins? What kind of actin, right? Okay. And how do formins and ARP differ? Formins bind to the plus end, actins bind to the actin. ARP binds to the minus end. Okay. What else? Is there any other differences? Are they involved with different types of processes, structures, anything? Yes.
Isn't ARP binded at an angle to make up a web of actin, whereas formins straight?
Yep, ARP binds at a 17-degree angle and is associated with formation of actin webs. And formins are associated, formins create straight polymers and are associated with creating contractile or bundles of actin. Yep. What else? Did we miss it? Did anyone else have anything that they put down that they want to share? So that's your substrates, how they work, kinds of structures and cellular functions they're associated with. So what kinds of structures and cellular functions are they associated with? This is creaking and bothering me. I'll just be listening to that the whole time. So there was something about ARP and angles and webs. What kind of cellular function is that involved with? Movement. Where would it be located in the cell? Periphery, leading edge, cortex, yeah. Foramens, what are they associated with? What kind of function? Also cell movement, but in a different way. Which, yep.
Isn't the foreman's more for cell division?
For cell division, so you mean that contractile ring, so more contractile actin. Yep. Yep. Also, we'll learn a little bit about it. So like cilia, and microvilli. Those are actin, parallel actin that are made by foramens. What about microtubules? What kinds of structures and functions are they associated with? Yep. Centrosome. So what's that? What's the centrosome involved with? In what? So starting with cell division, mitosis. In what way? How is the centrosome different in mitosis versus interphase? Duplicates, mitotic spindle. Great. Okay. Okay. Does anyone else want to add anything? Yes. So that's why I put a question mark, because I didn't know if that was, because substrate is usually what an enzyme works on, right? Here, foramens and ARP and gamma-tubulin, they work on specific type of protein, so I wanted to know actin or tubulin. Yeah, so that's why I went up the target protein. Okay. Good. So today, first let me check this one. So today I'm going to try something new. I've got this, and then if this PowerPoint starts going crazy, I've got it up on this guy over here. So hopefully, well, I'll be good. But I'm not going to play any movies on my laptop, so that should minimize chaos. Sadly, despite talking to OTS over and over again and being told they're doing this or that for me, nobody seems to have any ability to follow up on what they say they're going to do. It's really frustrating. Like, oh, we need to get you more memory. Tell some of your serial number, and it's not. Anywho. Okay, so far, this is what we've done. This is what you need to be studying. Obviously, plus today and Thursday stuff. Key players, major concepts, toxins and homologs, and then we started getting into the accessory proteins, which you can start categorizing as nucleating proteins. So that's your gamma-tubulin ring complex, ARP and WASP are the foramens. Subunit proteins that bind subunits and affect their activity or availability. Staphmin, profilin, thymosin, and that's where we stopped last week. Oh, yes.
So WASP is also actin, right?
WASP is a cofactor for ARP and activates ARP with actin. Okay. And so remember last week's paper had WASP and ARP in it. So now we're on to a new category, severing proteins. What do you think severing proteins do? Yeah, sometimes it's so easy, right? Good. They sever. So they regulate if you want to get all scientific about it, they regulate the length and behavior of actin filaments and microtubules. And so this picture is showing microtubules that are severed by katanin, which is, I believe, sword in Japanese or something like that. I once had a Japanese student, though, and she looked at me funny when I said that, so I don't know. That's what I heard. But that's almost like when you go and get a tattoo that's like something in another language, and then you always wonder if the person it was like, this stupid person got this stupid tattoo. You think it means peace and love. Anywho, So like maybe in the textbook it says they're like Japanese for sword, and really like some Japanese postdoc is like, oh, it means sushi, or I don't know. I don't know. Anywho, it was embarrassing though, because I'm like, oh, katana, Japanese for sword, and the students like. So you guys can look that up. You can be your own Stump the Teacher that has no points attached to it. So in this picture, the red is microtubules. And in A, they added katanin for 30 seconds, and in B, they added katanin for 3 minutes. And you can see microtubules are kind of severed a little bit in 30 seconds, much more over a longer period of time. Makes sense, right? So why would we do this? So we need our microtubules. Why would we be severing them? Wait, because remember the whole dynamic nature of the cytoskeleton and how it has to respond to the needs of the cell. And so if you need to quickly disassemble your microtubules, when would you need to quickly disassemble your microtubules? When you're done with mitosis, perhaps, or maybe before mitosis, right? That transition from interphase to prophase, there's a lot of disassembly and reassembly that needs to happen. So katanin can accelerate assembly of new filaments or promote depolymerization of old filaments. Wait a minute. I just said katanin severs. So how is it accelerating assembly of new filaments? Yes? It's increasing the concentration of subunits. If you have more subunits, there's more subunits available to polymerize microtubules, right? Makes sense. So this plays an important role in meiosis and mitosis. And so as you've seen with all of these proteins we've been talking about, there's always a similar protein with a similar function for actin, and in this case it's called gelsulin. Questions about this? Okay, so that's it for severing proteins. So now we have a new category. Proteins that bind to the side of filaments. Okay. So these proteins, and I always get confusion here, so I'm just going to say this now. These proteins can have a stabilizing or destabilizing effect. So don't just say they do one or the other. You have to know which protein you're talking about. And so with microtubules, they're called MAPs, microtubule-associated proteins. It's a whole family of proteins. Some are stabilizing, some are destabilizing. This picture is showing in green a MAP protein called tau. This is a neuron. And so you've got your axon and your dendrites. And so you've got two different MAPs here, one called tau and one called MAP2, and they're in different locations in the neuron. Would you say that these MAPs are stabilizing or destabilizing based on what you know about, for example, an axon and microtubules in an axon? Stabilizing, right? You want that transport in the axon to not go away, right? So you want stable microtubules. So here are these two different MAP proteins. What kind of MAP proteins are these? These two are MAP proteins that bind to the side of microtubules, like all MAP proteins, and they project outwards, regulating the organization of microtubule bundles. Think of them as spacers. Do you guys ever lay tile? Tile anything? Who likes to do? Who flips houses? You guys don't flip houses? I just saw an old student who just graduated, and I was like, oh, so what are you doing? He's like, well, I'm just flipping houses right now. That's it. The way to, I mean, in this market, not a good idea, but anywho. So you put the spacers in between to make sure they all have their space. So think about these proteins as spacers. For those of you who haven't laid tiles, I apologize for the poor example. It's better than, I used to explain domains as modular, it's called modular, modular, whatever. Okay, so I'm not gonna explain my bad example to you. I'll not do that too. So this one has a long spacer and this one has a short spacer. So if you imagine there's other microtubules that are gonna be next to it. So imagine there's a bunch of them in a row. This one, the next one's gonna go over here. And for this one, the next one's gonna go here. And that's exactly how it looks, right? So this is a cross section here. And each little circle is a microtubule. Notice how they're equally spaced? This is gonna have a longer spacer like MAP2, and this is going to have a shorter spacer like tau. And so why do they need to be spaced differently? Well, because the cell needs to be organized in a way so it can function as it needs to function. And so by keeping microtubules organized in certain cells, this allows the cell to function how it needs to function. Yes?
So is tau like always shorter than MAP2's?
Tau has this, it's the graphic designer's rendering of tau, but it is always smaller than MAP2. Yeah. So these are not examples. They're examples of MAP proteins, but the structures are kind of a good estimation of the effect it would have on the microtubule. Okay, questions? Yes.
Yeah, the previous slide said that they disassembled.
No.
Sorry, destabilized.
No, so I want to make sure that you understand destabilize or stabilize. MAP proteins in general are a whole family of proteins, microtubule associated proteins. That's all that means. So depending on the protein, it'll either be stabilizing or destabilizing. These proteins are stabilizing and are associated with organization and bundling of microtubules in a regular conformation. Yeah, that's why I'm glad you raised your hand and asked that, because every single year people think MAP proteins are one protein that does one thing, and I just want everyone to understand whole family, many proteins, and you'll see some more. Yes. Yes.
You put that tau is associated with Alzheimer's. Are you saying that it is responsible for irregularities?
Tau is associated with Alzheimer's. So tau is irregular in Alzheimer's. Yeah. I'm trying to remember. I actually didn't brush up on that particular aspect this morning. I'm spacing on the exact.
Stop the teacher.
Well, so it shouldn't stop me. That's why it's bothersome. I believe it's abnormal, so you end up with abnormal microtubule spacing, and so abnormal neuron function. It's really embarrassing, only because I worked on a system that was related to this for my PhD. So I'll have to get back to you on that. We're going to get a really good stump to teacher soon, but half of these I can mostly answer, and then I tell you to go out, and you basically give me back what I say. So MAP proteins are regulated by kinases. You have to think about that for a minute. So I told you MAP proteins can either stabilize or destabilize microtubules. They're regulated by kinases. What does that mean? Yes? Do MAP proteins themselves have SH2 domains? I do not know, but I will ask you a question. If I say they're regulated by kinases, what do you think is happening to MAP proteins? They're being phosphorylated. So, and I know they're always whispering. Can you tell them to stop? Yeah, no, so they're phosphorylated. So, to correct, just change what you had asked before a little bit, you would say, do they bind to proteins with SH2 domains? Probably. Or, maybe they don't, because SH2 domains is often associated with signaling. There's different types of phosphorylation that just does different kinds of things. What if the ability of MAP proteins to bind to microtubules is regulated by that phosphorylation? right? Think of it that way. So let's say destabilizing protein can only bind when not phosphorylated. It can only bind to microtubules when not phosphorylated. And so in the beginning of cell cycle, like, or the beginning of M phase, perhaps, you would have phosphorylation or dephosphorylation of that protein in order to allow it to then go bind to and destabilize microtubules to help produce the supplement, for example. So they're controlled actually by cyclin-dependent kinases, CDKs, which you've probably heard before, and you'll hear a lot more of in the next few weeks. Right, so just think for a minute. I want you to think about what this really means, because I feel like over the semester you guys have gotten really good at thinking about things deeply, and so I'm going to give you another opportunity to do that. MAP proteins are regulated by kinases. What do you think the effect of this regulation might be on cell cycle, especially the fact that they're regulated by CDKs? What does that mean? Because if you're studying and all you do is go regulated by kinases, you'll have like this much knowledge, but if you think about what that means, you'll have like that much knowledge. Does anyone want to tell me what they think regulation of MAP proteins by CDKs might mean? Yep.
Does it help with preventing cancer? So like make sure cell division is regulated and not divided by one. That's just my guess.
I'm just trying to think about where you're coming from. So does phosphorylation of MAP proteins, is that involved with cancer? Because you want to make sure cell division is not happening all the time. Okay. So I see where you're coming from. It wasn't, you went like, woo, which is good. Because it's a different way to think about it, but CDKs have many targets. CDKs are kinases that are activated specifically in specific phases of the cell cycle, and they phosphorylate many, many things. And so one could speculate that when that CDK is not active at a different phase of the cell cycle, then certain MAP proteins would not be active, and that would prevent perhaps disassembly or assembly of mitotic signal. Is that kind of where you were going? Okay, I'll go with it. Logical. Yep. And oh my God, two of you next to each other. Okay, go.
Would you say like it's kind of a checkpoint, not checkpoint per se, like how we study checkpoint, but like a checkpoint for cell division, as in, let's say the MAP is not stabilized. The microtubules needs to be disassociated, so you kind of would make sure that happens before you move on to the next step.
So it's a form of regulation of cell division. Your microtubules need to be disassociated before you can proceed with mitotic spindle formation. Or, yeah. So this would be a way to regulate that. Yeah. Did you have something you want to add?
Yes, checkpoint regulation.
Yeah, I would caution using the word checkpoint. But definitely in terms of regulation of proceeding through the cell cycle. Yeah.
So does the phosphorylation have to do with whether they're stabilizing or destabilizing?
No. The question one says phosphorylation have to do with whether MAP proteins are stabilizing or destabilizing. No, it has to do with whether or not MAP proteins are able to bind to microtubules. Different MAP proteins are going to, let's say there are 10 different types of MAP proteins, two of which we just talked about. Let's say there are 50 of them. Some of them are going to be destabilizing, some will be stabilizing.
And
That's,
activated by the phosphorylation.
and the phosphorylation is whether or not they can bind to microtubules or not.
Yeah, so that was.
Any other questions? I saw something back here. Yes.
What?
It was kind of a little bit related to what you said. She was saying that maybe phosphorylation of MAP proteins during cell cycle is a way to kind of regulate proceeding through cell cycle, because if a requirement for mitotic spindle assembly is disassembly of microtubules, then if that kinase is not active, if this CDK is not active, you can't phosphorylate, you can't activate, and so on. Yeah. Okay, thank you for that exercise of deep thinking. Yes?
Are they involved with like spacing chromosomes too in the line?
Are they?
Like say like when you're going through cell cycle dividing chromosomes line up, if like spindle tagging they regulate like how far they separate from one another, so they separate properly.
Do MAP proteins regulate chromosome segregation? Yes, actually, because I believe kinesin, so kinesin 13, is it destabilizing that protein? Which I'm not 100%, but it's logical to assume that because what they're really doing is regulating microtubule length, right? In this case of assembly disassembly, you can either have it regulating spacing, like what we just told you, or regulating length and stability. And so I can see how regulation of MAP proteins there could control when chromosome segregation begins. Other thoughts, questions? Okay. Okay, that was fun. Okay, so let's talk about actin. So we talked about MAP proteins with microtubules. I gave you, there are a few examples of MAP proteins. We'll have a few more coming up soon. With actin, there's cofilin. This is a protein that binds to the side of actin. And what cofilin does is it binds to the side and twists the actin filament, and that basically weakens the contacts between the subunits and destabilizes. So basically breaks down actin filaments into subunits. Interestingly, cofilin prefers to bind to ADP-bound actin. So it's prone to dissociation of old actin filaments. How did I know they were old? Because they bind to ATP-bound actin. ATP is newly added. ADP is older. Because once you add your actin subunit to your polymer, you get ATP hydrolysis. So it's almost like a little age. I don't know, like counting rings on a tree, right? You know the age of your actin. This is also interesting later on as we talk about movement and ARP and how it creates these webs of actin pushing out the leading edge of the cell. Then behind you have cofilin. So you have all this new actin polymerizing in the direction that the cell is moving, and then in the rear you have cofilin binding, disassembling the old actin, creating a new pool of subunits that can then be added to the front so you can keep moving forward. Very cool. Yes?
I don't really talk about it, but I think in the book I saw that actin
Yes.
So
So actin, you can add to the plus end and the minus end. It's just that you're more likely to add. So your rate of addition is much greater at the plus end than at the minus end. And your rate of dissociation is much greater at the minus end than at the plus end.
still fine.
Cofilin would most likely be binding to. Yep. Okay, questions? And so what you were referring to there about addition to the minus end, that is very much when you're talking about actin dynamics in a test tube, and all you have are just actin subunits, and it's all governed by critical concentration. And the cell with all these proteins that are coming into play, capping proteins, which we're going to be learning about shortly, that kind of control which enzymes can be added to the process. Okay, so then there's tropomyosin. This is another side-binding protein for actin. And it's stabilizing. So cofilin was destabilizing. Tropomyosin is stabilizing. Who's heard of tropomyosin? Good. Of course you did, because you had to take AMP, and a lot of you are taking some sort of other physiology course. What's tropomyosin involved in? Does it make sense that it's stabilizing, knowing what it does? Would it be weird if I told you it was destabilizing? Would you believe me? Never believe the professor. The professor is half crazy.
That's
I know. You guys are going to leave here and be like, tropomyosin is destabilizing. No, stabilizing. Tropomyosin also can prevent actin, interaction of actin with other proteins. Dysregulation is an important step in muscle contraction. And when we talk about that, you guys will get to take a brain breather because you all know it already. I always love teaching muscle contraction because you guys have always learned it in greater depth. And so when you hear it from me, everyone has this look of relief on their face. So that's good, I hope. Okay, so let's do some practice problems to liven things up. Actin filaments in a test tube behave differently than those in a cell. They assemble more readily and have a longer half-life. Why? Provide examples of mechanisms contributing to this observation. So take a minute to talk with your friends, and I'll circulate and explain the problem if you're confused, and then we'll discuss in about 2 minutes or so.
What stops them? Yeah, no, what stops it? Well, what makes it stable? So, what? No, stable. No, destabilizing. That's to do with how but in the test tube, we saw that how or it would be called root sticker application.
That's why it's faster. That's what well, so let's talk about that together. So, okay, guys, let's quickly go over the first one. Active filaments in a test tube behave differently than those in a cell. They assemble more readily and have a longer half-life. You need to think about there's two things. One is they assemble more readily. Two is that they have a longer half-life. Why? So what was the answer people had for why? Yes?
We got something to the covalent.
So I just heard it has something to do with cofilin. That's one accessory protein that might have one effect, but go more broad than that. Yes?
You don't have thymosin taking away and sequestering some of the monomers of actin?
You don't have thymosin. Thymosin should have any effect on actin? Yes, it should. Right. So I was just thinking about microtubules for a minute. I'm like, no, that's not. Okay, so you guys are getting very specific. Accessory proteins, right? If they assemble more readily and have a longer half-life, if you give me one accessory protein and one function, you will probably get it wrong because you're discounting all the rest of the accessory proteins. Every single one has an effect on half-life, right? And all the nucleating proteins have an effect on the fact they assemble more readily, right? So you want to think in test tube, we only have actin in the cell. We have actin in all these other proteins that we just went through. And all those proteins do is regulate assembly and stability, right? That's all they do, pretty much. So provide examples of mechanisms contributing to this observation. So the first observation is they assemble more readily. So what mechanism is behind the fact that they assemble more readily? I know, who said that? Nucleation, right? What proteins nucleate actin? Actin formants. Are they in your test tube? No, I just said actin filaments. I think on an exam I'll write by themselves to make sure it's very clear. But if you have actin by itself and you have no nucleating proteins, what does that growth curve look like? Right? You have that lag phase, right? When you have nucleating proteins, you lose that lag phase, so they assemble more readily. Yes?
So then isn't this question wrong to say that they assemble more readily?
Because the essential protein is not there.
Oh! I'm talking about it from the context, from the perspective of the cell, but in reality, it's worded from the perspective of the test tube.
Okay, that's right.
Yeah.
Okay, so
Now then,
let's turn it around. See, I told you the professors have crazy. No, exactly. Thank you so much for that clarification. So they're different in the test tube than there's in the cell. So what I would say is, in the cell, they assemble more readily and have a longer half-life.
But I think in the text it's different in the student has it.
how can it have,
Oh, well, you have to find that.
how can it have a long half-life in the cell?
Okay, so I just got the question, how does it have a longer half-life in the cell? Someone give me an answer. Why actin might have a longer half-life in the cell than in a test tube? Yes? Accessory protein, specifically which accessory protein that we learned about contributes to a longer half-life of an actin filament? Tropomyosin. Exactly. So where I say provide examples contributing of mechanisms, you would say, well, in the cell there's tropomyosin. Possibly in these cells there's high levels of tropomyosin activity. Maybe these are muscle cells. Like that. Yep.
If you were to say that in the cell it had a shorter half-life, could Jessalyn
Jolsolin.
Jessalyn?
So if I was to say in the cell there's a shorter half-life, could I say Jolsolin might be contributing to it? Yes. As long as you understand your categories of stabilizing and destabilizing, you can choose anything as long as it makes sense. Right? So if you told me Jolsolin contributes to a longer half-life, that would be wrong. But if it was the opposite, yeah. So we've got nucleating proteins and we've got stabilizing proteins such as tropomyosin. Those are present in the cell and would contribute to the fact that they assemble more readily and have a longer half-life compared to actin filaments alone in the test tube. Okay, so any questions about that? So if on the exam I give you this kind of question, raise your hand so I can clarify it. Don't just be like, I don't know it, because that's not going to be good for anyone. Cofilin preferentially binds ADP actin and is therefore more likely to disassemble older rather than newer actin filaments. Why? Would older filaments be at the minus end? Is that your question?
I said wouldn't they, so we want to disassemble them.
Well, that's kind of circular. You're having circular reasoning here. So I want you to tell me, so I'm telling you a fact. Cofilin binds preferentially to ADP-bound actin and is therefore more likely to disassemble older rather than newer actin filaments. Why? Yes? Exactly. Only hydrolyzed ATP is ADP, right? And only ATP bound actin can be added to a growing actin filament. But there's a little bit of a lag time. And after it's added, then you'll get ATP hydrolysis, and then it'll be ADP bound actin. So by definition, your older part of your actin filament is going to be ADP bound. You were there, you were just kind of dancing around it. Little dance. At least you were missing the addition of the lag. Okay. Who wants to explain that answer? Sometimes it's easier when a student does it. Or not, not the professor. I didn't sleep well last night. Child in my bed kicking me. Kids, you have kids, they come to bed with you, and then they kick you. So why is co-filament likely to disassemble older actin filaments? Why is ADP actin older? Yes?
Good try. So when you're adding subunits, they're going to be in the ATP bound state whenever you add them on. And then it takes time for like hydrolysis to occur. So when you have hydrolysis, it goes from ATP to ADP. And like I said, it takes time, so the new ones wouldn't have the ADP on them. It would be on the older ones.
Great. Did you get that?
I kind of feel like this, the question already answered this.
Yeah, you just need to understand that the whole thing's not ADP, but you need to be able to relate ADP to time. Or ATP hydrolysis to time. In order to
So
answer.
essentially, is that it's because fulfilling only bonds ADP actin.
Co-filament only binds ATP active, but you're not addressing that as older. See me later, I'm going to discuss it further. Okay, capping proteins. Yes, we're still going. Sensory proteins. So now we have capping proteins. These are proteins that bind to the ends of filaments and have greater effects on filament dynamics than those that bind to the sides. We just went over side binding proteins that either stabilize or destabilize. And now we have end binding proteins. So an example of CAP-Z. CAP-Z caps at the plus end of actin. Where's CAP-Z located? Anyone know? What kind of cell? Muscle cell? And it's stabilizing. And so it kind of makes sense. What happens is when you cap either end, plus, minus, both, and so muscle cells, you've got your plus end and your minus end capped. So are these actin filaments dynamic, or are they stable? Stable. You want them to be stable. If they were dynamic, we'd be in a lot of trouble. And so this is kind of just showing that without capping, you have this dynamic instability, and with capping, it's less dynamic. One molecule of capping protein has greater effects than one molecule of side binding protein. Why? What did you say?
I said because your filament grows in length, not in width.
Because your filament grows in length, not in width. That's close. That's good, but I feel like I want to hear more. Anyone? Well, so proteins that bind to, yep.
Well, if the filament grows at the minus end and you're capping the plus end, unless you have the
Severance.
severance protein, you won't have any like treadmill or anything. So it would be able to stay stabilized at the end that it would dissociate.
So by capping at one end, you're going to completely control length, right? Which is related to what you were saying. Whereas on the side, what are you able to control what are you controlling on the side? Just those few subunits that it's bound to. So let me go a little bit further, right? So if you think about these individual proteins we've talked about, you can think about it in terms of numbers of accessory protein that bind to numbers of subunits. So how many molecules of regulatory protein are required per molecule of actin or tubulin to have the desired effect? So tropomyosin binds to 7 actin subunits. So it can stabilize 7 actin subunits. Well, there's going to be a lot more than 7 subunits in an actin filament. So by capping it and preventing growth or shrinkage, you're going to stabilize the whole entire actin filament. Tau only binds to 4 tubulin subunits. Cofilin only binds to 1 actin subunit. So if you add cofilin, it's only going to take off 1 actin subunit from your filament. Whereas if you have a capping protein that somehow destabilizes, then it could affect the whole actin. Okay? And that's because that's where you're adding things to. If you're capping either end, you're going to lose that dynamic instability. Okay? So end-binding proteins are way more effective than side-binding proteins. In muscle cells, long-lived actin filaments are capped at the plus end with cap Z and at the minus end by tropomodulin. How does this affect actin filament dynamics? Does it stabilize or destabilize? I only said it on the Okay, good. So here's these more MAP proteins I told you about. These are proteins that bind to microtubule ends. Are also called MAPs because those are microtubule associated proteins. They bind along the side, but at the end. And they influence the frequency of catastrophe and rescues. So you've got, for example, a MAP protein called XMAP215, which stabilizes, and this is phosphorylated and inhibited during mitosis. Yes? Yes, because it's binding to the end. Yeah. MAP proteins that bind to the end, or you've also got your proteins that bind to the end of actin, and that's the capping proteins. Yeah, they're all kind of capping proteins, but where it gets confusing is they too are microtubule associated proteins, and so they share. That's what happens when people discover proteins. They just number them, and so some scientists said microtubule associated protein 1, MAP 1, MAP 2, MAP 3, MAP 4, and before you know it, you've got a million MAPs. Then there's catastrophe factors, and what they do is they destabilize. There are also microtubule-associated proteins that bind to the end, but they're catastrophe factors, so it's impossible to forget what that does, right? It's a catastrophe factor, and so it increases the frequency of catastrophes. And so you could think about it as there's this competition between stabilizing MAPs and destabilizing MAPs, or also known as catastrophe factors. That's kind of controlling microtubule length depending on where you are in cell cycle and the needs of the cell. How's it going? No one's sleeping. That's good. No one's crying. I used to always have someone who sit like right in the middle and just be like, that's the worst. Okay, so now we've got plus tips. So these are also end-binding proteins, but they bind to the plus end. So that's why they're called plus tips. Plus end tracking proteins. And this is kind of cool because they bind to the plus ends of microtubules and dissociate when the microtubules shrink. So you can imagine if you fluorescently labeled your plus tips and you did live cell imaging, what would you see? Let's say you labeled them green for fun, or red, so it's like this. Which direction would you see these plus tip proteins moving in your cell? From where to where? I'm going to tell you again. People always answer my questions, so we're going to try that one. Plus tips. These proteins, magical proteins that bind to microtubules, but they dissociate when the microtubules are shrinking, such as catastrophe. If you had fluorescently labeled plus tip, where would you see, and then you did live cell imaging, so you were looking at cells that were alive and following that fluorescence. Where would you see, what direction would you see those plus tips moving in? To the periphery of the cell. From where? From the center. Because where would they start? The centrosome, right? Microtubules grow from the centrosome outwards. Plus tips only bind to growing microtubules, so they're going to move outwards. They dissociate when the microtubules are shrinking, so you wouldn't see them moving back in. Does that make sense to people? There are other so there are catastrophe factors. Catastrophe factors in XMAP 215. So these are the ones I spoke about. These are plus tips because they are these plus end tracking proteins. They bind to the plus end and bind only moving outwards, and they modulate protein shrinkage. There are other plus tips. One is called EB1, and it's involved in microtubule positioning, and it's important for the microtubule, yeast microtubule spindle positioning. Is it important that your mitotic spindle is positioned properly, or do you think it could just be anywhere in the cell? It's pretty important, because if they're like, if one's like in the middle of the cell and the other one's over there, and then you go to segregate, how is that even going to work? It's very important that the mitotic spindle is at the opposite poles of the cell, right? Okay. In order for the cytoskeletal filaments to be useful, they need to be organized and attached to a cell structure. So that's the bulk of our accessory proteins. And now we need to, so now you understand microtubule dynamics, you understand those basic concepts, nucleation, and concentration, and subunits and polymers, you understand all of that. Now you understand all these different proteins that help to regulate your dynamics. And so what, right? Well, these structures need to participate in different cellular functions. But for that to happen, they need to be organized and anchored to certain places in the cell. So the example of that is the centrosome, right? All your microtubules are anchored to your centrosome. And things can kind of move along there. If you just had random microtubules, not anchored to anything in the cell, how would that serve any function, right? I feel like I lost half the class just now. She likes to think about microtubules and how they should not just be floating around. Like when you're going for a run in the morning. Well, in the bubble bath at night. Okay. You guys can go away. I lost y'all.